Today I'm extremely pleased to welcome and introduce my good friend Michael Goodisman to give his talk on social evolution in insects. Mike's been with us at Georgia Tech in the School of Biological Sciences for 16 years. He arrived here as an assistant professor and has been with us all the way through. His BA was from Cornell, and then he received his Ph.D. from that college up the road, UGA, with Marjorie Asmussen and Ken Ross, during which time he was recipient of an EPA Star Fellowship. He then saw some sort of light and went and did a postdoc in Australia, geneticist at Melbourne and Townsville for a couple of years, and then spent 3 years as an NIH postdoc, so remarkably both NSF and NIH postdocs in Tucson, Arizona with Dana Wheeler and Mike Wells. Then he came to Tech, as I mentioned, I think in 2004. He has 70 papers with at least 10 citations, so very productive output, during which time he's established himself really as one of the world experts in epigenetics and social insects. Much of that work done in collaboration with people like Sujin Yi and several others here at Tech. So he's been a very important member of our department in the evolutionary group. He's a dedicated advisor to graduate students and a mentor to undergraduates, and we're extremely happy to have him actively involved in the study abroad program. So he directs the Pacific program currently, if that ever gets back up and going, if Australia opens its borders, and also in Valencia in Spain. He's played, as we all know, a critical role in the school's administration during the transition, the merger with applied physiology, and his service to the department, the school, and the community is absolutely exemplary. And appropriately enough, Mike works, among other insects, on yellow jackets, which has given him some prominence here at Georgia Tech. Also work on fire ants has been profiled by the BBC, among others. And his abstract for today's talk is an absolute smorgasbord of topics in epigenetics and social evolution. And so without further ado, I'll hand it over to you, Mike.
Thank you very much, Greg. Hopefully you can all hear me. If not, so be it. Okay, let us begin. So I will be telling you today about social evolution in insects. So I'm going to start off with kind of a big picture view of the world and tell you that biological complexity, as we think about it, has increased through several major evolutionary transitions whereby individuals came together to form efficient groups. And I'm talking about things like the formation of chromosomes from unlinked replicating molecules, formation of sexual populations from asexual clones, and even multicellularity. And one of the most recent major evolutionary transitions was when solitary individuals, more or less solitary individuals, came together to form colonies or societies that were extremely interdependent. And again, this was kind of a coalescing of individuals into a higher level group. And my research is kind of focused on understanding this one of the last major evolutionary transitions. I'm interested in the causes and consequences of sociality. And I use genetic, behavioral, physical, computational, and molecular approaches to understand the evolution of sociality. And I primarily study sociality in the insects, the social insects, some of which are shown here. So who are the social insects? Extremely briefly. Primarily, these are ants, termites, and social bees and social wasps, and they've been extremely successful ecologically, and they form these highly interdependent societies. The reproductive caste cannot live without the workers. The workers cannot reproduce. These are kind of a fundamental group that cannot exist without each other. The real kind of defining feature of an advanced society is a caste system, especially a reproductive caste system, whereby some individuals reproduce and some, like these termite workers, do not, or typically do not, or like these honeybee workers, they cannot or do not reproduce. And so insect societies in particular have become models for studying sociality. So today I'm going to tell you about two branches of my research in the past several years. The first having to do with epigenetics in social insects, and the second, something completely different, having to do with sort of evolutionary conflict theory in insect societies. So first, I'm going to tell you again about some work in epigenetics that we have done in the past several years. So these insect societies, and in general social species, again, their defining feature is the caste system. And in order to have reproductive castes, you have to have some kind of developmental plasticity, phenotypic plasticity, okay? And that's because generally the casts are genetically, if we're talking about female casts, are genetically identical, but phenotypically different. Queens and males, which are not genetically identical, are specialized for reproduction. Workers and soldiers maintain the colony, but the castes that sort of define colonies differ dramatically in form and function. For example, here in this bottom panel, we see fire ants. The leftmost individual is the male. Next to the male is a queen, and then you have 3 workers that are all different sizes. The queen and the workers are basically genetically identical, but they look really completely different. And if you look over here to the termites, you see this big giant sausage. This thing is a termite queen. Around her are a few kings and workers, and you wouldn't even know that the queen is the same species as the worker if you didn't find them together in the nest. They look so different. And yet they are genetically identical, some of the workers to the queens. And so we have this issue of how do you get from one genome to multiple phenotypes? And the answer is that whenever you have sort of a genome producing different phenotypes, you have to have some kind of epigenetic information involved. And here I'm defining epigenetic information, epigenetics, as heritable changes in gene function not due to DNA sequence differences. And so this is kind of a molecular view of the world, that you have some molecular information that influences gene expression that causes ultimately the queen and worker castes to have different phenotypes. And when we think about epigenetic information, we're learning a lot about it. We think about things like histone modifications, shown in this figure, this complicated figure. You have different modifications to histone proteins that may carry information. You have different types of RNA, maybe long non-coding RNAs, maybe small RNAs that sort of can be inherited epigenetically. And you also have DNA methylation, shown by these little spikes. And it's really in DNA methylation that we have been focusing. So DNA methylation is one of the most common and best understood epigenetic marks. And when we think about DNA methylation in animals, we're usually thinking about the methylation of a cytosine base from cytosine to 5-methylcytosine. And the addition of this cytosine base imparts some kind of information that the cell can use to make decisions about gene expression. Now, DNA methylation a decade ago, a little more, was surprisingly, it had been extensively, not surprisingly, extensively well studied in mammalian systems, but was largely unknown in insects, largely because Drosophila, the model insect species, does not have DNA methylation. And so when DNA methylation was discovered in honeybees, it was a huge splash, a really important finding. And it was especially important because there were some papers that came out and said that DNA methylation may be important to caste formation. And so many folks became interested in this question, is DNA methylation the key to the development of castes and societies? And it's been a highly controversial subject in the past several years as people start to learn about what it is and what it's not. And so we have been studying the function of DNA methylation on insects for many years, and I'll give credit to the folks from my lab who actually did the work. Here, Brendan Hunt, who's currently a professor at the University of Georgia, Carl Glassad, currently, I think, still at the University of Pennsylvania as a postdoc, and Carl Dyson, who's still working with us here at Georgia Tech. And so one of the studies that we did quite a few years back is we kind of looked at patterns of DNA methylation within a social insect. And we specifically looked in termites, Zootermopsis nevadensis, if those folks are interested. And I'll just say briefly that termites have really interesting caste determination. So you have an egg here that develops into a larva. And again, these insects don't have a real larval stage. They have young that look a lot like the adults in complete metamorphosis. And then you have a sort of another kind of final, quote, larval instar. And then you can have differentiation happen into a full-on alate caste. These are winged individuals that will go and fly off and found new colonies. You can have differentiation into the soldier caste, which is highly modified head. You can have differentiation into a so-called secondary reproductive, or you can kind of have things just kind of stay the way they are. And so castes are complicated and interesting in termites. And so we sequenced the methylome of several termite castes in order to understand the patterns of these insects, and we found some interesting results. So what you see here on the left-hand side is the frequency of methylated cytosines in CpG dinucleotides in various parts of the genome for our termite, shown in green, and 2 other species for comparison. And the first thing that I'll point out to you is if we just look at this green bar in termites, most methylation is targeted towards exons, and some to introns, but little to sort of intergenic regions. And this is interesting because it's different from what we see in mammalian systems. The other thing that will probably jump out at you is that the levels of methylation in termites are much higher than they are in this ant, shown in blue, and the honeybee, shown in red. And I'll just tell you that termites diverged from ants and bees 350 million odd years ago. And for comparison, I think humans and whales diverged 100 million years ago. So there's a massive amount of evolution we're talking about when we look at these comparisons. So we took kind of a closer look at what methylation looked like within genes, and you're seeing here some scaffolds of the termite genome, along with levels of fractional methylation here shown by these peaks. And I'm just going to, you can sort of just see here, and I'll point out these little blue bars are, exons and genes as per normal. And again, you can really see that methylation targeted towards high gene-rich regions. Areas which don't have a lot of genes show very little methylation at all. And if we kind of blow up this section and squeeze in on this region of high methylation, we again see that our individual genes, shown by these kind of fishbone diagrams, they're pretty highly methylated. But again, these areas in front, quote, 5' or wherever in front of the genes show little methylation. And this is interesting because this pattern kind of looks a little bit like what we see in mammals, where we also have promoter regions, for example, being unmethylated, and the exons and introns being heavily methylated. Now, in mammals, we have much higher methylation in intergenic regions and transpose elements, but we do see some similarities between termites and mammalian systems. So we then looked at sort of expression. How are genes expressed as a function of methylation? And so what you see here are groups of genes of increasing methylation, and here you see a measure of gene expression. And you'll see that as methylation increases for a gene, expression also increases. Okay, so we have this positive relationship between DNA methylation and expression. But I'll point out some details in this figure. The trend is not super, it's kind of weird. It's not just a straight linear trend. In fact, it's the intermediately methylated genes that kind of have the highest expression. And as you kind of tail off down here, the most highly methylated genes have a little bit lower expression. And you might say, well, this is, maybe this is just a statistical artifact or something. But in fact, if you look at other species, you see the same trend. It's a kind of curved linear trend. It's not just a straight linear relationship. So there's something interesting going on there. We also were very interested in what was the relationship between DNA methylation levels and variance in gene expression. For example, are highly methylated genes differentially expressed, or vice versa? And what we found was kind of the opposite. If, again, we look here at DNA methylation level, increasing DNA methylation is negatively correlated with variance in expression. So that means that genes that are differentially expressed tend to be unmethylated. And this may sound a little different to those of you who study methylation in mammals, again, where variance expression is sometimes correlated with variance in promoter methylation. But here we kind of see a negative relationship between DNA methylation and variance in expression between castes, between sexes, between tissues, you name it, you kind of find this relationship. We also looked at differentially methylated genes between castes, and here we were looking at our alate versus non-reproductive castes, and here males versus females, and the number of differentially methylated genes and where those genes were more highly methylated. And the take-home message here is that we found a lot of differentially methylated genes. Where they are differentially methylated is interesting, maybe on the specifics, but it's actually controversial still whether differential methylation kind of happens at all. There's been some papers put out there kind of calling into question whether studies of differential methylation in insects have actually found it or whether it's statistical artifacts. We have found it, and other folks have as well now. And there are lots of differentially methylated genes. But again, what this is doing, we're still not sure of. Okay, so we've kind of studied the patterns of methylation, and the correlations with expression. We were also interested in studying how does DNA methylation affect gene duplication and vice versa. And this is an interesting topic to us and who study kind of complex phenotypes for a lot of reasons. Here you see an example of something that could be important. This is a fossilized wasp here from a long time ago. And you can imagine that way, way, way back, this wasp was solitary, did all of the functions of both reproductive and worker, and went about her business. But over time, we developed this caste system, as shown here again by these yellow jacket cats. Here we see the queen and worker. And these two castes do different functions. And so there's kind of an intriguing idea that gene duplication may be one way to separate functions. So maybe you have this gene A way back 100 million years ago, and then you have a duplication event into an A1 and A2, and this A1 gene does some kind of queen function. It doesn't have to be concerned about, having a worker function. This gene A2 does some kind of worker function. So we become interested in this question of gene duplication. And so we looked at patterns of gene duplication across the bees. So this is a phylogeny of bees. And this bar over here shows different levels of sociality defined in certain ways. And so the lightest, as you get lighter, you get the most complex societies, if you will, and the darker castes are solitary or mostly solitary. And what was interesting is we found a correlation between gene duplication and sociality, which is interesting to us. We also looked at patterns of gene expression as a function of gene duplication, and we found that duplicate genes, as shown in this first panel, for example, tended to show more caste-biased expression than singletons. And this again fits into this hypothesis that gene duplication, again, these duplicate genes show caste-biased expression, they may be co-opted into caste-biased phenotypes. The three different panels here are from three different data sets. They all show the same thing. And I'll also note we see the same thing with sex-biased expression and with tissue-biased expression. So biased expression is correlated with gene duplication. Be interesting. And I'll just kind of show this graph. It's not from our papers, but from a commentary on our paper, nicely showing this idea that gene duplication may be correlated with the evolution of social complexity. I will just, to be very clear, this is still very much a hypothesis. We found some evidence for it. Again, as we in this graph, we have one measure of complex sociality, increasing colony size as a measure of duplication rate. There's a slight correlation there. We need a lot more data, but it is an intriguing hypothesis. Now, regardless of that, we were interested in studying patterns of methylation amongst these duplicate genes, okay? And so what you see here are the percent methylation as a function of gene family size. So 1 is a singleton gene, 2 is duplicates, and 3 are triplicates or more. And you see this very, very strong correlation, that methylation is higher in singletons and then goes down quite abruptly in duplicated genes, and then in higher family sizes even more. So we do have this pattern of increased methylation for singletons and loss of methylation for duplicates. We then sort of looked at related duplicate genes, orthologs or paralogs in this case, and we were sort of looking at the difference in methylation between gene families. And so what you see here is the difference in percent methylation, and this big sort of distribution is our kind of null hypothesis based on randomization of singletons. And what we found is that pairs of duplicates showed less divergence in methylation, and maybe that's not surprising. Still interesting, I think, that duplicated genes tend to have relatively similar patterns of DNA methylation. That may make sense. We also did some fine sort of tuning on patterns of DNA methylation and gene duplication, and I want you to focus on the second set of bars, duplicates here. This first set of bars that says singletons, this is again a control set to just kind of make sure that our test was working properly. But what you see here are duplicates that were either on the same chromosome in honeybees or on different chromosomes, again in honeybees. And you see that duplicates that happen to end up on the same chromosome, probably by some tandem duplication event, as opposed to those on different chromosomes that may have been transposed or something, show differences in methylation. And so we do see that those are on the same chromosome tend to have similar levels of methylation, lower divergence. And again, this may make sense. There's something about the environment, the chromosomal environment, that keeps the methylation the same, or there's something about being moved to another chromosome that causes a divergence in methylation in interesting patterns. So I'm going to summarize this first part of my talk to say that what we have found is that patterns of DNA methylation are conserved among insects. And what I mean by that is that insects that have DNA methylation, we find the same pattern, that methylation is targeted to genes, to exons especially. That methylation is correlated with increased expression. That methylation is usually negatively correlated with variance in expression. Now, there's one big caveat in here that I'm just going to have to leave for now, which is that DNA methylation levels among insects vary dramatically. In the termite, we have high levels of DNA methylation, relatively high levels, but in Drosophila, we have essentially none. In the paper wasp, we have none, but in the honeybee, we have some. And there's very interesting patterns between species that we're still working through. I mentioned this, that methylated genes tend to be highly and uniformly expressed. And we've also found that DNA methylation is associated with gene duplication, although we're not sure what that means exactly, if that's a kind of a correlate of the other functions that we've seen. And this most important question, that some insects have DNA methylation and some don't, which raises the kind of big evolutionary question. If methylation is so important, why can some insects get away with not having it? Or why haven't they all lost it? Or why have some kept it? And what's going on here? And so that's going to lead me to this really great conclusion that I'm sure you were waiting for, which is that the function of DNA methylation in insects remains unclear. So it's very controversial about whether it causes change in expression. There are some folks out there who say it is not causal. There are experiments that show it's not causal, at least not simply causal. There are other folks who are now coming out with studies that say that there is causation, that a direct increase in methylation does cause an increase in expression. There's questions about, is this important to sociality? Well, that seems to be, now that's probably a no. So our original question that brought us in was DNA methylation the cause of advanced societies? It seems that's not the case. So that is one thing that we might be able to answer. And the big question about, what this is doing is still a great area of study for us and for many other groups out there. Okay, so we can exhale for just a second now. And in the next hour of my talk, I'm going to tell you about a completely different area of research that we've been very interested in for quite some time. Doesn't have to do with molecular biology and sort of different phenotypes, but has to do with evolutionary conflict, so more of an ecology and evolution focus. So I started off by telling you that insect societies are models of cooperation. Okay? We see that honeybees working together and swarming, ants retrieving food, murder hornets defending their colony with great vigor and gusto. But it turns out that societies also display conflict. Conflict between queens and workers, conflict between male mates, conflict between multiple queens in a nest. And this conflict comes about because individuals within colonies are not, they're not clonal. If they were clonal, then there would be no evolutionary conflict. You'd have no selection for conflict, really. But because they're not identical genetically, you have the opportunity for conflict. And this conflict can be quite severe. For example, quite famously, honeybee queens, when they are sort of born and when they're going to hopefully go on to have a new colony, they have some sisters that are potentially being reared next to them, and they will go and kill all their sisters off because their sisters represent competition. They actually sting through the cell wall or go into, well, I would say hand-to-hand combat, so that only in the end there is only one that remains. And there's also other kinds of interesting conflicts potentially between male mates. And so we've become very interested in this question of evolutionary conflict. And I'm going to tell you about some areas of research that we've done in this conflict question. And again, I need to give credit to the folks from my group who actually did the work. Jen Kovacs, who's currently a professor at Agnes Scott, Lynn Chow, who was at Minnesota but now is employed as a computational biologist, Carl, again, who's with us, and Eric Hoffman, who's a professor at the University of Central Florida. And so the first type of conflict I'm going to talk about arises from multiple mating. So multiple mating, also known as polyandry, is an interesting evolutionary question. In general, it's interesting. It's very interesting in societies. And so it turns out that there are some species of social insects that mate once, and there are some that mate multiply. Multiple mating would seem to be weird and bad for societies because multiple mating by queens will decrease nestmate relatedness. And we think of relatedness as being the sort of key thing that you want to keep high in order to keep societies together. But there may be benefits to multiple mating that we don't know about, but multiple mating can definitely cause different kinds of conflicts amongst individuals in societies. And these just show some species of multiple mating individuals. The honeybee famously mates with 20 to 30 males. Honeybee queen mates multiply. This is a harvester ant that mates with 5 to 10 males. And this is our good friend the yellow jacket that turns out also mates multiply. And I'm going to talk a little bit about the yellow jacket research we did just for fun. It has turned out to be a great model for studying a variety of evolutionary and ecological questions, and we're certainly still working on it. So there's interesting things we've learned about cooperation, conflict, and invasiveness, which I'll get to in just a few minutes. So we've been studying two species of yellow jackets, and again, this is so you learn a little bit of yellow jacket biology here. There are two common species of yellow jackets in the Atlanta area and in the southeast in general, Vespula maculifrons and Vespula squamosa. And Vespula is the yellow jacket family. There's about 20 to 25 species worldwide. Some of them are extremely damaging invasive species. From our perspective, the natural history of the two species is pretty similar. Basically, new colonies are founded by single queens who are responsible for all the reproduction that happens in the colony. One interesting side note I won't have a chance to talk about much now is that Vespula squamosa, which is shown here, is a social parasite of Vespula maculifrons, which that has a certain meaning in the human world, of course, but in the social insect world, it means that queens of this species, Vespula squamosa, invade established nests of this species and take it over, basically knock out the resident maculifrons queen, take over her nest, and take over the already established worker functions. Won't go into that much. Squamosa, also so you know, it's got these yellow racing stripes, so if you ever see that, watch out because the squamosa sting, much more painful than the maculifron sting. They're larger. They also tend to swarm a lot more. When they sting you, they drop a pheromone on you that attracts other wasps to hit you in the same place. So the squamosa are nastier than the maculifrons. You can ask my students about that. So in terms of kinds of methods, I'll just tell you that we've been using kind of classical genetic markers, microsatellite markers, to study patterns of relatedness and reproduction in Vespula societies. Suffice it to say that these markers allow us to say this worker was produced by this male, this queen is related to this worker. We can kind of determine genetic relationships. The table down here, again, not so relevant, just to let you know that the markers that we use for most of the work I'm going to tell you, they're highly variable. This is the number of observed alleles, in each species, lots of alleles, high number of effective alleles, high levels of heterozygosity. So we've got a lot of power to detect genetic lineages within colonies. That's pretty much all we need to talk about for the methods. So I'll say the first thing we found is that both species that we were studying do mate multiply. What you see here is the number of male mates of individual queens. So for example, here we found two Vespula maculifrons queens, that mated 3 times, and 2 that mated 4 times, etc., etc. The blue bars being for maculifrons, the eastern yellow jacket, so use its common name. The red bars being for squamosa, the southern yellow jacket. You'll notice that there's a shift in mate number. So although they all mate multiply, there's no queens that mate with 2 or fewer males from either species. We do see squamosa mating with more males, at least 6, whereas it seems like maculifrons mates with, a minimum of 3. And so we do see this pattern of multiple mating, which again to us is very interesting and causes potential conflict issues. So the first type of conflict I want to talk to you about that occurs when you have multiple mating is a conflict of who gets to reproduce, and specifically, which caste do males contribute to. So I'm showing you here a diagram of a single queen, shown in yellow, that is mated with 2 hypothetical males in red and blue. And in order to produce workers, it requires both an egg and, of course, sperm from the males. And I should just note that the mating, kind of interestingly, could spend a lot of time talking about this, but mating in social insects and in yellow jackets and honeybees happens all at one time. So there's a single day or days when the queen goes out, mates with 2, 3, 4, 15, 20 males, whatever, she stores that sperm and reproduces for the rest of her days just using that stored sperm, which is a whole interesting physiological problem unto itself. The males die off here, so they're not in the colonies. When I show this diagram, I'm just telling you who the father was. They have long died off, and I'm just kind of showing their genes being passed on. So what you see here is that the male red has contributed to 5 out of the 6 workers, has produced 5 out of the 6 workers. His sperm has been used for 5/6 of the workers. Male blue, only 1/6. And so that's interesting in and of itself. There's some kind of reproductive skew, as we call it, between the males. One male is getting more reproductive success than the other. That's important. Now you might say, okay, male red is the winner here. He's got 5/6 of the workers, and that's good for him, evolutionarily speaking. But you have to remember that workers are sterile, or mostly sterile, and will not typically reproduce. And so let's also say that we were looking at the same colony here, and yes, male red has gotten 5/6 of the workers, but if we look at the production of new queens that are produced, male red has only got, in this case, 1/4. Male blue has got 3/4. So who's the winner here? Well, good for male red for having produced all these workers. Evolutionary dead end. Male blue has kind of won this game by producing more of the reproductives. And so we went out to test whether there were differences in the proportions of reproduction, as we might see if there was conflict, the proportion of production of workers and queens, new queens, in Vespula maculifrons. And so this graph shows the results of that. Each sort of number here is ness 34, ness 35, ness 36. W and G means the workers. For W, G gynes means a pre-reproductive queen. You can just think about it as new queens. And each little sort of colored bar here is a particular male and his contribution to workers and to the gynes within that nest. So for example, in nest 34, it looks like we had 1, 2, 3, 4, 5, 6, 7, 8 males that mated with that queen. And you can see male 1, shown by this leftmost color pattern, contributed almost identically to workers and to queens. And male 2 was pretty much the same, and on and on and on. And if you do a test, kind of the p-value here, you do not find significant differences between the proportions of reproductive success to workers and gynes. And in fact, we didn't find that for any of the nests we looked at. And so we do not see evidence of this switching of reproductive success. So if you contribute a lot to workers as a male, you also contribute a lot of gynes, and we don't see the kind of cheating in terms of switching of castes that you might see if there was this kind of weird conflict going on. The next kind of conflict I want to talk to you about is conflict over sex ratio. So sex ratio, we think about mammalian sex ratios 50-50. What's, nothing too exciting about that if we think about transmission of the Y chromosome. But sex ratio can actually, and is actually, subject to selection in all kinds of interesting ways. And sex ratio in social insect colonies, and specifically I will say the ants, bees, and wasps, the hymenopteran insects, is of great interest because the optimal sex investment ratio for queens differs from the workers she produces. Okay? So
do you have like different people?
I'm getting someone helping with my talk. I appreciate that. So the sex ratio of queen wants is actually 3-to-1 investment in females, and that's because of her relative relatedness to her daughter and male offspring. The sex investment ratio that workers want is 1-to-1 of new queens to males. And so there's conflict between queens and males over sex investment ratio. What's interesting is that as queen mate number increases, if you have multiple mating increase, the relative relatedness asymmetry changes, and you get this kind of declining function so that at the asymptote, the sex investment ratio, ideally, for both for both workers and queens is the same. And so there's even some folks who think that this is why multiple mating evolved, because you get this decrease in conflict. So we were interested in looking at the sex investment ratios in Vespula maculifrons to see whether they matched the kind of predictions. What is the actual sex investment ratio? And I'll show you, I only don't have much time for it, what kind of relationship we found. And I'll tell you here, the x-axis is weird because I'm showing you worker relatedness as a decreasing function, and I'll show you why that is in a second. And I'm down showing you the sex investment ratio, which is queens to males. And for this slide, what I want to just show you is we do get kind of a relationship between queen mate number, which is a decreasing function of worker relatedness, okay, and sex investment ratio. So we do see a correlation between those two things. However, the correlation, of course, with all our research here, we never get exactly what you expect, but that makes it fun, I guess, right? So sex investment ratio does not match the predictions. I've now shown kind of, again, these two graphs, the theoretical expectation and the actual observed, as both functions of increasing queen mate number. First of all, although you may not notice it immediately, the sex investment ratio as a function of queens to males is less than 1, as shown here, okay, for most of the colonies we looked at. In principle, the sex investment ratio theoretically should never be below 1 for either queen or worker control. The other thing, which is more obvious, is that the sex investment ratio, investment maculifrons, it increases as queen mate number increases, which is not as what we would predict based on the kin selection theory. So, interesting, but did not match our predictions under regular conflict. So then we asked, okay, so what's going on here? Why do we have multiple mating happening? It doesn't seem to be happening because it's kind of matching this conflict theory. Maybe there's something else that multiple mating is doing. And so we asked, why do queens mate multiply? And we tried to look at some fitness correlates beyond the sex ratio. And one of the things that yellow jackets do for us, which is quite nice, is that they rear their young in cells, kind of like honeybee cells. You see here so-called cells that are used to rear workers. And I'll tell you that the yellow jacket comb is made of paper as opposed to wax, but otherwise very similar. And then you see here on the left, comb that is used to rear new queens. And we can basically go into a mature nest, pull it out at the end of the year, count cells, queen cells and worker cells, and get some kind of a correlation with fitness. We can determine how many individuals how many workers this individual produced, more or less, relatively speaking. How many new queens did it produce? And we can kind of see does that correlate with mate number. And the answer is it kind of does in Vespula maculifrons. Again, a very dirty relationship here, marginally significant, so do what you will with it. But what we see here is queen effective mate number and a function of the number of queen cells. And I'll tell you, if we looked at the same relationship as a function of worker cells, we do not see a correlation. And this is not a linear relationship, but I drew a line, just a trend line, so you can see it. We kind of have something weird going on. If you look at queens that mate effectively less than 4 times, these 6 colonies over here, they produce very, very few queen cells, with the exception of this one colony that did produce a substantial number. But then when you look at colonies that produced, where the queens mated more than 4 times, we have a great variance in the number of queen cells produced. We have some colonies that, don't produce a lot of queen cells. We have some that produce a whole bunch. So there could be something going on here. Okay, the last thing I'm going to talk to you about here has to do, again, with conflict in a different kind of Vespula yellow jacket nest. So first I have to tell you a little bit about the life cycle of yellow jackets, something you should all know being here at Georgia Tech, if you don't know already. So new yellow jacket colonies are actually founded by single queens when they hibernate, when they come out of hibernation after winter. So in this kind of life cycle wheel, we see a new queen initiate a colony, often at the base of a bush or somewhere in the ground, typically around here. And then she initially kind of builds the first little nest. She's the one who goes out and forages for food. She lays some eggs, and she rears up that first cohort of workers. The workers then take over the process of food collection and colony defense. And sort of through the spring and the summer, the colony grows. It gets bigger and bigger and bigger. So by the time you get to the autumn, actually right about now, you've got the fully mature nest, which looks something like this. You can see there's probably maybe 5 to 10 in a large colony of these combs, hundreds of thousands of worker cells that have been produced, hundreds to few thousand queen cells that may have been produced as the growth, is kind of exponential. And again, here we see a typical sized nest in the ground that's been dug up. So this is the typical cycle continues, and then right in the autumn, now what happens is the colony transitions from producing workers to producing new queens. Okay, so they pretty much cease producing new workers, kind of like a plant that kind of grows vegetatively and then transitions to producing flowers at a certain time of year. The flowers here, as I like to think of them, are the new queens and males. And so you'll have queen cell construction now, rearing of new queens, rearing of new males happening pretty much, now. We often think of Halloween as the kind of time of year when the colonies are chock-a-block with new queens and males. And it's just around that time, Halloween into Thanksgiving, when we see Vespula mating flights. So the new queens and males that have been produced fly off, they mate with each other, and then those queens will overwinter. They will hibernate in our relatively cold Georgia winter. All of the males will die off. All of the old workers will die off. All of the old queens will die off. And so that during the winter, when it's cold here, the entire population of yellow jackets is represented just by the queens that are hibernating somewhere and the sperm that they've collected through mating. So this is a typical annual life cycle. But what we see now more and more is changes to the life cycle. We see changes because of global warming. We see changes in invasive populations in Australia, New Zealand, South Africa, other places. And what happens in these warmer climates, whether it's Australia or whether it's South Georgia or South Alabama, is that the colony does not die off during the autumn and instead persists. And it keeps growing exponentially, and it persists and persists through the winter, and so at least some of the queen stays home, and the colony keeps on going. And so these are perennial nests that are doing something weird and something wonderful, I would say. These perennial nests can take on huge size very quickly. These are photos of Vespula squamosa, most likely perennial nests. Here's someone standing presumably next to a sprayed out nest of squamosa. You can see it's as larger than he is. This is probably maybe 2-3 years old. Here you see the inside of a car that has been completely taken over by a squamosa colony. Not sure exactly how this happens. It didn't drive it much, I guess. And the most spectacular example I've seen, this is a photo from New Zealand, it's different species, but the same idea, an older figure. Down here you see a scientist or someone wearing his uniform, yellow jacket uniform. The perennial nest here is about 15 feet high, maybe 5 feet across, and it's just kind of taken over this tree. And so these perennial nests are fascinating to us for a variety of reasons. They're interesting because they sort of represent changes in sort of society structure. They're interesting to us because they're correlated with, we're finding more and more of them in South Alabama and South Georgia because of rising temperatures. And so we were interested in studying these, and we collaborated with some folks at Auburn to study perennial nests in Vespula squamosa. And so the first thing we did is we looked at the relatedness of individuals within these perennial colonies. And what you see here are estimates of nestmate relatedness. This first bar is the estimate of relatedness among workers in annual colonies of the species. So our typical single queen annual colonies. And you can see it's relatively high, about 0.37. It's lower than the value of 0.75 you would expect if you had single mating by the queens, but I'll remind those of you who are kind of familiar with this that multiple mating by queens drops that relatedness down quite a bit. Regardless, from this graph, you can see it's relatively high. The relatedness of workers, of new queens that we find, and of males within these perennial supercolonies is much lower. Okay, so this is already interesting. Something is happening where there's got to be some kind of gene flow into these perennial colonies. It's probably not just, the same queen that's hanging out and producing all those workers. There's something else going on here. And one of those something else's is that we have found, again, by genetic analysis, that these supercolonies are headed by multiple queens. And this is important because most typically these Vespula colonies are headed by a single queen that dominates reproduction. Perennial colonies take on multiple queens, which then leads to possible conflict between these queens, but also certainly has something to do with the fact that these things can get so large. So again, we may have conflicting evolutionary processes here. So we were very interested in this relatedness issue. What's going on here with, we see this lowering of relatedness, what's happening? And so we decided to test that a little further by looking at the mitochondrial genotypes of workers within colonies. And what you see here are the sort of patterns representing, each different pattern represents a different mitochondrial haplotype, okay? So we have, in this population, we have the blue striped haplotype, we have the purple checkered haplotype, we have the red striped haplotype, and the yellow haplotype. Each circle and number represents a colony. So colony 2, as we're calling it, all the workers we sampled had that blue haplotype. Colony 6, all the workers we sampled and had the red haplotype. Now, this makes sense because if these annual colonies are headed by one single queen, then all the workers should inherit her mitochondrial haplotype. So there's nothing particularly exciting about this finding from annual colonies. What will be interesting is if we look in perennial colonies, because if we have the workers showing multiple mitochondrial haplotypes, that is going to tell us that the new queens that we are seeing and the workers potentially, or depending what's going on, that are in that colony, they are produced by non-nestmate queens. So we might have a queen coming in from outside and reproducing, and that's again something it's kind of a socially parasitic thing to do, and would be interesting. I mean, it's interesting either way. And so the question is, what did we find when we looked at the mitochondrial haplotypes in these perennial supercolonies? And the answer is that we found pretty much the same thing as we found in annual colonies. The vast majority, 99% of the individuals, all had the same haplotype within colonies. So this perennial supercolony HA, all individuals had the red haplotype. The perennial supercolony KL all had the same haplotype. We found only one individual in this colony MA that did not match, and of course finding one individual is quite annoying. We don't know if that's anything interesting. It could just be some kind of a mistake, but whatever it is, the overriding pattern is that we see only one mitochondrial haplotype within each colony. I'll also make one other kind of important point here, which you can see a little bit, but we did more sophisticated tests. We do not see genetic differences between the perennial supercolonies and the annual colonies. We looked at the nuclear genetic data as well. So there's an interesting question about whether these supercolonies might have some genetic component that allows them to become that. Are they a new type of species or incipient species? We see no evidence of that, although we really will have to do some genomics, and we will do some genomics to figure that out if that's really true or not. So to wrap up the second part of my talk, I'll say in our studies of cooperation and conflict in Vespula societies, we found that queens mate with multiple males, and we found some evidence that there are direct benefits to multiple mating for the queen. Again, we saw that increase in cell number, and other folks have also found kind of direct benefits. There's some evidence that in yellow jacket nests, higher mating is correlated with better disease resistance. We see that in honeybees as well. So we do have some evidence that multiple mating has benefits that single mating does not, which is interesting. We found that the patterns of sex ratio and offspring production were not consistent with kin conflict theory. That may partially be because you may or may not be able to have your cake and eat it too, if you will. You have some direct benefits going on, but the kin conflict can't play out. We're still not sure what's going on with sex ratio there. I think always more study is needed to see if that pattern is really holding and what are the correlates of sex ratio, but it was not exactly as we expected. And then these super colonies are very interesting to us, as we said. They seem like they're places for increased risk of conflict, but we haven't seen evidence for that quite yet, because we do have them still being kind of loose families. All of the queens are sisters, I guess, within there. But they may be selected for in these warmer climates. And so we're going to see more and more of them, and hopefully one day they'll make it all the way up to Atlanta. Not yet so far, but I'm certainly keeping my fingers crossed. And so to end, I'll say that we're continuing to study sociality, the causes and consequences of sociality, using all these different approaches. We hope to continue to do so in the future. And I will then end by thanking all of the folks who were heavily involved in the work. Size of font indicates some level of differential importance. And the funding agencies who have intermittently seen fit to fund me, thank you to you, and I'll thank you, the audience, for taking the time to listen, and I guess I'll take any questions if you have any.
Okay, thank you very much, Mike. I'm assuming there's a lot of virtual applause going on. It's very interesting stuff.
Yeah.
Maybe I can start by asking if you would speculate on, or maybe even have real hard data on linking the two parts of your talk. So what are the differences in methylation? Are you looking at differences in methylation among the castes, and do they change on the same time scale as the evolution of these supercolonies?
So, yeah, good question. So we have not looked at that, but we probably will. My feeling is that the interesting thing about methylation is that we're finding that these patterns of methylation is that these highly expressed genes and genes that do not show differential expression are methylated, and it turns out that it's the same genes across species that tend to be methylated. So if I told you that a particular gene was methylated in the honeybee, whatever it is, alcohol dehydrogenase, we'll just pick randomly, it is likely that that gene will also show the same methylation patterns in termites and in ants. And so the patterns of methylation seem to show conservation at levels of high species levels, I will say. And I didn't have time to talk too much about that, but that's very interesting. So to answer your question, we would love to look at differential methylation, between these social forms, and maybe we will, but we have not. And I don't know that we would find differential methylation between them, or at least not correlated with expression, but it's definitely interesting to do. Any questions in the chat? Okay.
May I ask a question?
Yes.
The question: have you ever looked into comparing the RNA transcriptome with the genome? If you have, for example, differences in RNA editing among the different castes in the colony?
Yeah, really good question. So we have not done that, but other folks have, and so there is differential expression of genes between castes, which is interesting. There is also differential splicing of some genes between castes, which is important. And to sort of go on with your question, there are some folks who think that methylation, we know that methylation can affect splicing in some species. There's excellent evidence in plants, at least in other places. There's also correlations between differential splicing and DNA methylation in insects. So that's another area, yes, that's really interesting and could be another function of DNA methylation. Questions?
Yuri popped up. You got a question here?
Yeah. So I actually have two questions. One, in continuation on Francesca's question, did anyone you or somebody else look at proteome differences? Are differences in protein modification, solubility levels, and some other parameters between castes?
So the answer to that is no. There is very little protein work done. I think, obviously, as you know, genetic genes are easier than protein work. And so, the level of molecular biology, there's a few labs that have done a little bit of differential protein analysis at a very crude level. And you find differential proteins, of course, between castes. But the proteome is not well studied.
And another question is to second part of your talk. So when you're dealing with these perennial colonies, which do not die at winter, does it mean that the lifespan is changing? The lifespan of workers and especially lifespan of queens?
A very good question. Does the lifespan of queens and workers change? We don't know. So it's a very interesting question because there's big differences in lifespans between castes in general. This is one of the other fascinating things. A fire ant worker will probably live in the field maybe 3 months or so. A fire ant queen can survive 7 years, and there are termite queens that can survive decades potentially. And so my prediction would be that with sort of increased crowding and things like that, you get a lowering of lifespan overall. But we do not know. We don't know how to aid. We actually had a conversation about this the other day in the lab, how to age workers and queens in the lab. We know from studies in fire ants that multiple queen fire ant colonies, the queens do live less, maybe only a year or so. And so there is something about being in these crowded societies with lots of reproductives that seems less good to the individuals in terms of lifespan.
So, Mike, since we're going around the ohms, what about the microbiome? I mean, there must be a big pressure on infectious disease, in a sense, in these supercolonies.
Yes, microbiome is very, very interesting too. All these great, interesting things. So there are some groups studying the microbiome in honeybees, and social insect colonies and societies in general are thought to be subject to heavy infectious pressure. Yellow jackets, living underground in the dirt, and fire ants, lots of bad microbes. There are lots of, we think, antimicrobial secretions that ants and termites have. There are folks who have studied the microbiome in the honeybee, like looking at our microbiome to see if it's related with health, and it is. There was a study that just came out. We're not doing the microbiome so much, although I'd love to do so. But there are interesting things going on, both with the microbiome that's trying to take out the societies versus that is used by insects. And we know that in aphids, for example, there's microbes or now obligate endosymbionts that are critical for survival and function. Doesn't seem to be quite that case in honeybees, but there is definitely important microbes that they have.
All right. Well, that brings us right up to the top of the hour. We've got a number of congratulations comments in the chat. So without any further questions, I guess we can stop the recording, Jasmine. And thank you all. And again, we'll see you next week in hybrid mode.
Thank you all for coming.
Thanks a lot, Mike.